Good morning to all the folks tuning in and about to be presenting from Hawaii. Good afternoon to everyone on the East Coast, and good evening to folks tuning in from Southeast Asia, India. It's really great to have everybody together. This will be our last BioacousTalks session of 2024. All the prior talks that we've done are on our Yang Center YouTube channel. We can send a link in the chat. And like, if you imagine this year as a concert, this is a nice headliner to go out on because we are welcoming two researchers, Amanda Nevine and Pat Hart, who together with Tom Denton and Matt Weldy have been working on a new approach for measuring the density of Hawaiian birds. And why does that matter? Well, if you can hear from Pat Hart's house, you can hear bird calls and some late remnants of the dawn chorus, but they're from mynas and from yellow-fronted canaries, none of the native birds we were listening to. And so Pat and Amanda and others at the LOHE Lab and elsewhere are working to use bioacoustics to understand this decline in Hawaiian birds and then really apply these approaches to support them and bring them back. And so with that, we're gonna turn it over to Pat, who's then going to hand over the floor to Amanda. And we have a Q&A set up down below, where if you have any questions at some time during the presentation, just put them in the Q&A chat and we'll be able to have some great time for questions at the end of the presentation. So Pat and the canaries and mynas, I'm handing you over the floor now.
Thank you. Mahalo, Ben. And thank you so much, everyone, for coming today. I'm amazed to see so many people from so many places. Makes me nervous. Anyways, I just really want to thank my co-authors, especially Amanda Nevine and also Tom Denton, who I think is here today. Matt Weldy from Oregon State has been working with us a lot, and also our friend Rick Camp, who I didn't put on the talk for certain reasons, for political reasons. He works with the federal government. They need to have everything okayed first. But anyways, Rick has been a really important collaborator for us too. So what I want to do is start off just for the first 5 or 10 minutes, giving a quick intro to our situation in Hawaii, a little bit about Hawaiian birds and why we've been doing this work, and then hand it over to Amanda, who's going to talk about the nuts and bolts of this process of how we're working on estimating, basically the creation of this new variable called density that Amanda and Tom have been integral in creating, and then also how we can use that to estimate the abundance of birds and their density. So thank you. I will move forward. Let me know, feel free to ask any questions. So Hawaii, is really well known around the world for having just this incredible diversity of birds, most of which have evolved from a single ancestor that arrived here over 7 million years ago, and just evolved in isolation over that time in the absence of mammals, reptiles, amphibians, and a lot of insects that you find around the world. And so because the Hawaiian birds evolved in isolation, they lost their resistance to a lot of diseases that birds anywhere else in the world are resistant to. And so in the early 1800s, a whaling ship visiting Maui, there was an accidental release of a mosquito, Culex quinquefasciatus. Mosquitoes had never been in Hawaii before. These are vectors for avian malaria. And within 100 years, we started losing a lot of our diversity of forest birds. The Hawaiian honeycreepers are a really well-known radiation of the birds. There was about 56 species and subspecies of them before humans and mosquitoes got here. We're down to about 17 species of these birds now. And so most of our birds are living in remnant high elevation forests. Our islands are really high. This is Mauna Kea, which gets about 4,000 meters elevation, close to 14,000 feet. The mosquitoes are a tropical mosquito. They mostly live in the lowlands. And so because of that, we have few native birds in the lowlands, and most of our birds are in these higher elevation forests, but they're limited by going higher by tree line around here. And with climate change, the mosquitoes are moving up in elevation. And so it looks like we've lost an additional 2 of our 17 species on the island of Kauai just in the last couple years, really. So there's been a lot of work focusing on landscape scale mosquito control so we can save our Hawaiian birds. The incompatible insect technique is one of them. Basically, all insects have Wolbachia bacteria in their gut. It turns out if you mate a mosquito with one strain of Wolbachia with the strain of mosquito with the mosquito with a different strain, the eggs will be infertile. And so there's been a big effort over the last 2 years to drop Wolbachia infected mosquitoes across the landscape to control the wild mosquitoes. You get the males with this different strain of Wolbachia to mate with the females, the wild females with a different strain to make them sterile. And so there's a huge amount of money. This is a really expensive project. We really treasure our Hawaiian birds here. They're our remaining native birds. We want to do everything we can to preserve them. And so, there's a lot of effort going into this. And so as our populations are becoming smaller, we need better information about how many there are left. We need to be able to monitor them more efficiently than we currently are, and also know where they are, and so that we can make these management efforts more successful. And so, the method that's been used even up till today is the point transect method. Basically, you set up transects through the forest, and at stations along the transect, you stop, and for 8 minutes, you record every bird that you see and hear, and especially you look at the distance to each bird, and then you use that information, you plug that information to the program distance, which some of you may have used, and you can model, basically, after a bit of work, you can model the density of birds over the area that you've surveyed. Yeah. And this is really, we call it, the gold standard for estimating bird density and abundance right now. The problem with it, is that it's really hard to find enough trained observers. You got to know the songs and calls of every species. In Hawaii in particular, our birds are really variable in what they say to each other. They have each species has a lot of different songs and calls. We have a lot of rare species. And so, if you spend 8 minutes on a point listening for the birds, you might miss any rare species in that area, right? It's hard to detect the distance in dense forest to the bird, and the distance to the bird is really important, is a really important measure in estimating the density. A lot of our forests are super remote and difficult to access. You have to have helicopters to get into a lot of our remaining Hawaiian wet forests. There's very few roads, and it's the landscape is very difficult to walk through. And finally, estimates based on a single 8-minute count at each station. If you do that once per year or once every 5 years, it's not the best way to estimate the number of birds in an area, right? And so, as I think all of you know that are here today, sound is the way that birds keep track of themselves. It's the way they keep track of who's around, how many of them are around, and where they all are. And so I think many of you here today are probably working on projects or know people working on projects using passive acoustic monitoring. And it has great potential, to improve the way that we can estimate the distribution and abundance of birds, because you can record for hours and months and days, and it's just a massive amount of data that can give us a lot of good information about how many birds there are if we only have the tools to analyze it properly. And so, just for fun, I think this is the dawn chorus at Hakalau. This is an area, our birds are declining throughout the state, but we do have places where the birds are still doing super well and where they're at really high densities. And Hakalau is one of them. And I'll just play a quick little soundscape just so you can hear what that sounds like. And I think Amanda's going to be coming on soon, and you'll probably hear a bit more of that. And so we started, because we have such an extinction crisis in Hawaii, we have been working on ways to improve the way we can monitor birds for a long time, especially using sound. We've had passive acoustic recorders out in some forests for 10 years now, recording continuously. And we've been working on, trying to use these soundscape data to improve the way we can monitor the birds. A few years back, I was working with a colleague, Esther Sebastian Gonzalez, and we worked on a way to use the sound pressure level from passive acoustic recordings to estimate the distance to birds, and then getting density that way. It turns out there's problems with that method, in that it seems like in Hawaii, especially, some sounds like the ones that we used for that, the birds do keep the pressure level similar. It's a particular call, but on a lot of songs that the birds sing, they seem to modulate the sound pressure level. So it's difficult to use that sound pressure level to estimate distance, because there's not a good relationship between the distance of the bird and the sound pressure level at the receiver. So we've been working on other ways to improve that. Collaborating, meeting Tom Denton about a year and a half ago has really helped our lab just, expand exponentially what we've been able to accomplish over the last year and a half to improve the way we can monitor birds. And so anyways, we've been producing terabytes of data, trying to figure out a way to analyze this data. And so this is where I'm going to pass over the talk to Amanda Naveen, who is our bioacoustics informatics technician in the LOHE Lab. She's been with us for the last 5 years, and she's going to talk about basically the work that has come out in 2 papers that's been put out by us and Tom over the last year, and how that relates to improving the way we can monitor distribution abundance of birds moving forward. So, Amanda.
Hi, everybody. Good morning, afternoon, evening, depending on your time zone.
Can you get it? Can you share, Amanda?
I think you have to stop first.
I need to stop.
There you go. Make
Oops.
sure I share with audio as well. Share sound. Okay. Can you see
Got
that?
it?
Looks great.
Oh,
Okay.
okay. Thank you. Okay. So like Pat said, my name is Amanda Naveen. I'm a bioacoustic research specialist at the University of Hawai'i, and I'm going to describe our method that we've developed alongside Google's Bioacoustic Research Group to use bioacoustic data to estimate population densities, which we previously haven't really been able to do. Throughout my slides, you'll find this QR code at the bottom right corner. It will direct you to our most recent paper in ecological indicators, where you can read more about the details of our bioacoustic metric and our method for estimating it. So I'll just kind of glance over this because Pat did a great job of introducing it. But as we all know, we've made huge advances in passive acoustic monitoring over the last couple decades, both in terms of hardware for recording the soundscape and software for analyzing bioacoustic data. In particular, advances in machine learning algorithms for the semi-automated detection of vocalizing species within acoustic data, or classifiers, have made passive soundscape recording a really feasible approach for continuous wildlife monitoring by exponentially reducing our analysis time. So for instance, incorporating PAM-generated detection, non-detection data from classifiers into occupancy models has proven really effective for monitoring geographic trends in vocalizing species occurrence. However, translating this PAM data into more sensitive indicators like animal density, for example, remains really challenging. For this reason, passive acoustic monitoring still faces some skepticism from wildlife managers as being a robust and timely approach for accurately assessing important population metrics, such as absolute abundance and animal population density on the landscape. Various methods have been proposed for estimating absolute animal density from PAM data, each with some limitations. So many attempts to replicate, many have attempted to replicate traditional point count distance sampling. As Pat mentioned, distance sampling is conducted by trained observers who go into the field and count animals, and they estimate the horizontal distance to each of those detections. As Pat also mentioned, it is kind of the gold standard for population monitoring, especially here in Hawaii, where we've used it for a number of decades. To replicate this method using in a PAM setting, distances have to be estimated between an automated recording unit at each station or ARU and each individual vocalization of the birds in the landscape. Triangulation methods, which use time of detection and direction of arrival at multiple microphones to identify individuals, can be effective for this, but in order to use those methods, you have to have a really densely deployed grid of ARUs at each point, which can make it expensive to survey a relatively small area. Another approach that doesn't require discrimination between individuals involves estimating distance based on sound pressure levels, but as Pat described, there are some issues there as well, as the relationship between sound pressure levels and distance are influenced by weather, vocalization directionality of the bird, individual variation in vocalization intensity, etc. Further, simply manually calculating detection radii in the field is also complicated by the influence of environmental factors, which can vary across survey locations and conditions. Not to mention, it can just be very labor intensive if you have a really large grid of ARUs on the landscape. So alternative methods have been developed that bypass distance estimation from recordings altogether. One promising approach is to use vocalization rates within recordings, which has shown really strong correlations with traditional abundance estimates. So vocal activity rates, or VAR, as in the number of individuals or vocalizations per unit time, and Q rates, the number of vocalizations divided by the average rate at which individuals vocalize, have shown really great potential for tracking population trends. These methods assume that vocalization rates are density dependent, where the number of vocalization increases predictably with the number of individuals. However, temporal variation in vocal behavior can confound this assumption. VAR can be approximated from classifier prediction outputs using detection rates. So the proportion of short audio samples, typically 2 to 5 seconds long, within a PAM dataset triggering automated classifier detections above a selected classifier confidence score threshold. But classifier detections can be problematic due to trade-offs between false positives and false negative detection rates. Misdetections have to be accounted for before vocalization counts can be reliably used to track populations. Additionally, misdetection rates depend on the data distribution. Species-specific vocalization rates and acoustic detection probabilities vary across time and space, meaning confidence score thresholds may not transfer very well between datasets due to differences in species behavior, environmental conditions, or community composition of birds. So take, for instance, this variable classifier performance we see here across species and sites. It might be safe to pick a consistent confidence threshold across sites for species with consistent classifier performance, like Amakihi or Oma'o, but picking a threshold for Akea Pola'au based on performance at site 1 will certainly skew our estimates at site 4. What's more, classifier performance can vary pretty widely at different times of the year as species vocalization behaviors change between breeding season and non-breeding season. So calibrating our threshold selection based on breeding season false positive rates will likely yield wildly inaccurate estimates at other times of the year. Further, performance can vary between habitats and have different acoustic features. Pat showed that picture of that open landscape versus the really dense forest. So we can expect that those false positive rates are going to vary between those two sites, and in areas with different species assemblages as well. So take, for example, this variable performance we see for Alopaio. It varies quite widely, and precision may have been lower at certain sites if they also contain a species known for mimicry, the EEV. In addition to their own songs, EEV produce mimicries of other birds like Alopaio that can be tricky for even trained humans to tell apart. So here, have a listen to a true Alopaio call. Were you able to hear that? No?
No.
That's a little sad. Oh, it's playing on my screen. Well, oh well. Have a look at this true Alopaio spectrogram, and then have a look at the EEV. And you can see that they're really quite similar, and I apologize that the sound isn't coming through. But it's a hard enough task to discern these two calls from one another for me to do, let alone a machine, especially when you're only considering these really short clips like a machine learning classifier might. Lucky for us, EEV usually have a tell if given more than a few seconds. They can't help but sing their own song on top of their mimicry. But our classifier may not get access to those longer clips in order to find that tell. So our classifier may therefore assess these two spectrograms and, given only that information, score them both highly for Alopaio but low for EEV. So if we think about what the distribution of confidence scores for EEV might look like, presumably they're more distinctive call types, which sounds like a rusty hinge that I can't play for you right now. High on the confidence axis, while the Alopaio mimicry calls that they do are going to fall lower on that axis. If we're assuming that only audio samples with high classifier scores are true detections, we'll be dramatically undercounting the number of EEV calls in this data set. Further, if we then assess the same data set for Alopaio, we could likely dramatically overcount them if we assume that the mimics are all being classified by the classifier as Alopaio. So altogether, these challenges relating to the variation in acoustic detection probability and classifier performance really limit our ability to use PAM data to estimate sensitive indicators like animal density. But not to fret, we have a solution. The underlying species densities is a compelling target in this regard. The call density is the proportion of total audio samples assessed by the classifier that contain the target species localization. So when greater than 0, call density can serve as an occupancy indicator. And after establishing occupancy, changes in call density may also indicate changes in abundance or behavior or population health. The distinction between call density and classifier detection rate is subtle, but quite consequential. So the call density metric assesses several limitations of existing methods. Most notably, call density is not dependent on high classifier performance because it assesses the entire distribution of model outputs. And I'll get more into this in a minute. Because we assess the entire distribution of model outputs, we can account for a lot of variability in vocal behavior across time and space and detectability amongst sites. And thus, it's less reliant on consistent decision boundaries around arbitrary threshold levels. We don't have to fine-tune a threshold selection for each species or each site. Our method for estimating call density uses a fixed amount of human validation effort and is therefore readily scalable to large projects, and is applicable to be used with any classifier that assesses fixed time audio samples, which means you could use it to analyze output from Perch or BirdNet or YamNet, etc. So our equation to estimate call densities is based on the law of total probability and Bayesian reasoning, but at its simplest, it is the audio samples containing a target signal divided by the total number of samples. Because we expect the probability of true detections to vary across the distribution of confidence scores, and because we expect these distributions to shift between study sites and time of the year, we use a logarithmic binning scheme to convert the continuous classifier scores into discrete sets, or bins. Then we validate manually samples from within each bin to calculate the call densities across all bins. So I won't go into too much detail about this equation. If you want to learn about the math behind our method, please follow the QR code on this page to our earlier paper in Frontiers in Bird Science, or I'd be happy to discuss it later, but please direct any hard questions to Tom. Thank you. Instead, I will continue by describing the workflow we have developed for estimating call densities. I'm going to use this distribution of confidence scores generated by the Google Perch classifier for Amakihi, that cute little yellow guy there in the corner. So we're going to use our logarithmic binning scheme to convert the continuous classifier scores into discrete sets, such that the lowest scoring 50% of audio samples are assigned to the first bin, the next lowest 25 are assigned to bin 2, etc., so that the last bin gathers up the remaining highest scoring examples. With this design, we know exactly the probability that any sample chosen at random from the dataset falls into each bin, or the bin probability, which is the first part of our equation. We then validate a fixed number of samples within each bin, making a tally of true and false samples. The logarithmic binning focuses our validation effort on higher scoring samples in those upper two bins. And we would expect that if the classifier has a decent quality and the prevalence of the target species of low, this is really going to help us to find those elusive samples. However, that not to say that for rarer species, more validation of samples might be necessary. So validation across this entire distribution of scores gives us a chance to catch those tricky samples that might have been discarded if they fall below a threshold, such as low-scoring calls masked in the cacophony of the dawn chorus that may have otherwise been thrown out, or high-scoring false positives for mimicry or acoustically similar species. Samples perhaps like these, and again, I apologize that I don't seem to be able to play them for you, but if we're running an analysis for Amakihi, which make trills, these two samples may achieve similar scores and be assigned to the same bin. In a threshold-based analysis, they might both have been counted as a true Amakihi detection. However, see Doug has a hand raised.
We'll have time for questions at the end, or feel free to use the Q&A down below.
So they might have both been counted as true detections if we had used a threshold-based method and these had scored similarly. But the first sample at the top, we can discern in our validation protocol is in fact an Amakihi, whereas the second is actually the trill of an Alavi. And marking this false sample allows us to assess what proportion of samples might be similarly false within the bin. So we expect that the higher scoring bins will have a higher proportion of audio samples containing true detections, but this might not always be the case. So using these validation results, we can next model each bin with a beta distribution, with a small constant C representing the uninformative prior to estimate the probability that a sample selected at random from each bin is a true detection. This is the second part of our call density equation. We can then bootstrap sample from this beta distribution, plugging each value into our equation to get a distribution of call density estimates, which allows us to calculate the maximum likelihood estimate and construct confidence intervals. So we've ground truthed this method for generating call densities using both simulated and fully annotated acoustic datasets and found it to yield estimates that are robust to variation in call density and classifier model quality. And our next step was to assess the relationship between call densities and bird densities on the landscape. For our case study, we used 3 years of recordings synchronously collected alongside point counts, such that the two metrics, bird density and call density, were estimated at the exact same level. The 8 native forest bird species that we studied vary widely in population demographics, and our study sites varied from subalpine dry woodland to montane rainforest to test the applicability of our approach across different bird densities and ecotypes. To evaluate the call density indicator compared to other bioacoustic indicators, we also derived 2 threshold-based acoustic metrics, but I'll only describe the highest performing detection rate. Detection rate was calculated at 2 selected thresholds by multiplying the number of audio samples with scores exceeding the threshold by the likely true positive rates determined by manual validation, and dividing that by the total number of samples. The 2 thresholds we decided to assess were the top scoring 25% and 12.5% of samples, respectively, which corresponds to the 2 highest logarithmic bins from our call density protocol. So this way we could use the same set of validations for all bioacoustic metrics that we derived. To our delight, we found that for every bird species we studied, call densities are strongly correlated with bird densities on the landscape. These plots demonstrate the linear relationships between the maximum likelihood call density estimate on the X and the bird density estimated using point transect distance sampling on the Y. So each point in these plots represent an independent survey, and the whiskers represent the confidence intervals along the two density estimates. Despite the range of bird densities that we saw, call density was a strong predictor for each. And if we zoom in on our two rarest species, the Akiapola'au and the Akepa, who are consistently below 2 birds per hectare, we can see that our validation protocol can handle low densities. These two species also happened to have the highest classifier performance. They have pretty predictable vocalizations, and they both have ROC AUC scores around 0.9. Important to note that although the threshold-based metric detection rate performed well for most species, it was only a notably better predictor of bird density than call density for this rarest species, the Akiapola'au. This plot on the right has detection rate versus bird density, with the purple points indicating the top 25%. Those are the circles, the 25% threshold, and then the teal points, which are triangles, indicate the top 12.5% threshold. So this finding that detection rate is really effective for rare species with a good classifier aligns with previous studies that suggest that detection rate is potentially a really useful proxy for the number of individuals of a solitary species, but might be less indicative of group size in the case of gregarious species. The strong correlation between detection rate and bird density in this case might be due to the species' rarity and the classifier's high accuracy, which resulted in near perfect capture of the vocalizations within the top 25% of audio samples. As we can see by the number of overlapping points between the two thresholds, it seems like we were capturing most of their true vocalizations from within this data set, within this set of above this threshold. So this coincidentally created an ideal threshold for calculating detection rate. Where call density vastly outperformed threshold-based metrics was for species that form flocks and species that had poor classifier performance. The dense, highly vocal animal populations pose unique difficulties to the traditional monitoring schemes, and even more so for PAM approaches, where recordings can become saturated with continuous vocalizations. However, despite Apopane here on the left, nearly saturating recordings above 50 birds per hectare, call density actually remained a strong predictor of bird density. This could not be said for detection rate. We saw a very weak relationship between detection rate and bird density at both selected thresholds for Apopane, and we can see in the plot on the right that densities as low as 20 birds per hectare actually saturated the detection rate for the species, where all samples above the selected threshold contain their vocalizations. By not discarding the lower scoring samples, call density is actually able to buffer these saturation effects more effectively. So rather unsurprisingly, for birds with high population density, we actually found that we could improve bird density predictions by moving beyond linear regressions. Exponential or other models provide a much tighter fit, as we can see here for Apopane. But this is an area of future work. We want to really investigate what the best relationships are between call density and bird density, and looking forward to investigating that in the upcoming year. Call density also excels for species with poor classifier performance, as I mentioned. In our case study, EEV, the mimic, and Oma'o had the lowest classifier performances with ROC AUC scores under 0.7, but they still had strong relationships between call density and bird density, outperforming detection rate. And this is even before we did nonlinear model fitting. So altogether, our findings indicate that call density is more robust to variation in animal density, from dense Apopane to sparse Akiapola'au, and classifier performance from the mimicking EEV to the predictable Akepa, than threshold-based metrics like classifier detection rate. Further research is really needed to deduce if this method is similarly suitable for assessing other acoustically active species like anurans or cetaceans, though any species that can be assessed using distance sampling methods should theoretically be accessible with our method as well. As Pat mentioned, call density estimation has really revolutionized the way we're able to make ecological inference from acoustic data here in Hawaii. And we now have active PAM projects across all the main Hawaiian Islands, as well as some in the northwestern Hawaiian Islands. Because there's a fixed amount of required human validation, and we can estimate call density from a single recorder instead of a time-calibrated array, estimates for large and small-scale projects alike can be generated really rapidly. equipping wildlife managers with the information they need to make timely decisions for the conservation of threatened species. Though further research is needed, our case study suggests our method is applicable for a variety of species population demographics and ecotypes, and may be more indicative of changes in population health than detection, non-detection based analyses that might only detect species decline after local extirpation. So this project has been the result of many years of hard work from many individuals, but I want to thank in particular Tom Denton at Google, Matt Weldy at Oregon State, and Rick Camp at USGS, and of course my PI, Pat. I also want to thank all of the bioacoustic giants who came before me, whose work I was able to build upon here. Again, here's the QR code for our most recent paper describing this work in ecological indicators. But if you have any questions, I think I have time to take them now. Otherwise, I'm happy to respond to emails. So thank you for listening.
Wow, Amanda, Pat, thank you so much for this extremely informative and inspiring talk. I think we have several questions in the chat that I can read out. And then anybody else with questions, feel free to put it in the chat, or we can activate your microphones. So I think for the first question of is there a publication on the techniques that you've used to estimate density using the sound pressure level of bird calls? I do think Pat has a paper on that, and we can link it in the chat down
Yeah,
below.
that's Sebastian Gonzalez et al. 2018. I could send that if I can find it.
Perfect. So now let's go to Vijay. He is wondering, how does call density vary from vocal activity, assuming that the classifier performs well? Samples with audio or detections over total audio.
Yeah, so that's a great question. So oftentimes vocal activity rate is manually assessed, where recordings are scanned and annotations are made for each vocalization in order to calculate that vocal activity rate, which can be quite labor intensive for large quantities of data. So instead, vocal activity rate can be approximated using detection rate of the classifier. When a classifier performs really well and the clustering of samples is really tight, of true samples is really tight, then call density and vocal activity rate actually line up quite nicely. The difference comes in when the classifier performs poorly, or in certain other situations where call density's ability to account for all of the samples within the data set instead of just those that scored highly is where we see the difference, where call density is a little bit more robust than vocal activity rate.
Any follow-up on that, Vijay, or?
See the
You've
Yes,
got
I
the mic now,
don't know how
Vijay.
to unmute myself. Yeah. Uh, no, thanks so much, Amanda. This is really cool work. Yeah, I would love to read the two papers before I have follow-up questions. So I think I'll follow up with all of you via email. Yeah, amazing work.
and I thank you. And I see that your last comment asked about the code and the analysis. This GitHub, this Git repo for Perch is the link to the code for running the classifier in our analysis. And then also, if you follow the QR code to our paper, that will also link you to a Zenodo repository where we have some of the data from this study that you can access.
Awesome. Thank you so much.
Brilliant. Let's turn over to Nicole, who is wondering, how does the call density metric account for the fact that some species call much more frequently than others, say red-eye vireos versus a woodpecker? So even if they're both present at a point at the same abundance / density, but they have different calling rates, how do you account for that?
Let me back here. So when we build these relationships between call density and bird density, we can see that the slopes of them are quite different, where or can be quite different, where we are accounting for the variability in vocal behavior in that we're building these relationships on a per species basis. So we can say, okay, if we look at call density of any one of these individuals, at like 0.1, say, for instance, and we look at the relationship for all of we're looking at about 3 birds per hectare, but with Akiapola'au, we're looking at, about 1 at the same call density. So as we're conducting these analyses and building these relationships, we're considering that change in behavior for each species.
So does there still need to be some type of ground truthing where, if people are looking to apply this in a different area, would you recommend that they do some amount of, transect surveys, or some, basically like how you validated this approach?
Yeah,
Is that
yeah.
necessary?
Yeah, that
Yeah.
depends completely on your goals. So if you want to estimate density, bird density on the landscape using this method, then there's going to be some calibration steps necessary. You're going to have to conduct the ground truthing in order to get those estimates. However, we've shown that call density is a really good standalone indicator where inference made from call density and bird density would be the same as far as making decisions for management. So call density can be used independently, or it can be calibrated to give you these other population metrics.
Nice. And another question is, how does this approach, or does it address, and if so, how, some of the just fundamental problems of doing density analysis, like propagation environment changing throughout the year, or noise levels changing at different times of the year, some of that yearly variability, and site to site differences. I understand that how that could result in a change in the distribution of detections, and that this method accounts for that, but still I'm curious how it addresses those challenges. Yeah.
Yeah, so as you mentioned, the distribution shift in the data across time and across space is accounted for by our strategy of discretizing the confidence scores and then validating across the entire range of scores. As for the change in vocal behavior across time as between breeding season and non-breeding season, obviously you wouldn't want to compare one site's breeding season call density to another site's non-breeding call density and assume that that's going to have the same inference for population status. So it's important to compare apples to apples, right? You want to be looking at call densities from similar time windows across these different locations.
That makes sense. We've got a few questions related to like sampling schedules and duration one needs to record. So Bianca is wondering how much data should ideally be used to develop this type of model for a single species. And then I saw an earlier question about like how do subsampling schedules, with your recording schedule, how does that potentially affect the accuracy of this approach?
Yeah, so that's something we actually really are interested in exploring is whether or not using longer recording schedules or longer recordings in developing these relationships between bird density and call density would yield even tighter estimates. One of the cool parts about this metric is that it's really flexible where you can choose how you discretize your data and stratify it however best suits your study question. So for this project, we used 8-minute recordings, which correlated perfectly with the point counts that were conducted on the landscape. So the call densities were from a collection of 8-minute clips from a survey area. But you could, theoretically, you could have any recording schedule that you like, but you have to realize that if you are recording 24 hours, then your daytime vocalizing species are going to have, much lower call density because they're only vocalizing for a small portion of your recording schedule. So it's important to consider your study species of interest when you're designing your recording schedules. You definitely want to target their peak vocalization windows. So for instance, we recommend for birds, conducting your passive acoustic monitoring during the same windows when we would typically be conducting point counts. So we conduct point counts from dawn until about 11. Here in Hawaii, that is the peak vocalization window for most of our species, and then that would align nicely for our PAM schedule. Did I answer that full question? I think there was a couple of facets to that one. Sure.
I guess one question I have is, what's next? Are there ways in which you're looking to improve this approach, or yeah, I'm curious what you're planning on doing with either refining this method or scaling it. Yeah.
There's so many things we want to do with this, but I see that Tom wants to have some input here.
Tom, let's give you the mic. Where are you, Tom? Tommy, can you hear me? Okay. Yeah, I'm allowing you to talk. Let's go.
There we go. Okay, cool. Now I think I'm unmuted.
Yep.
Yeah, sorry, I just wanted to throw in first, thanks, Pat and Amanda, for taking in a rogue like me. And it's been great working with you. And it's really exciting problem space that y'all are working in for so many reasons. So I think one of the things I wanted to emphasize that I think speaks a little bit to a couple of the earlier questions, but also to the future work is, I think there's a lot of questions around how covariates and call density interact, and exploring, how do we integrate covariates in reasonable ways to say, like, okay, is it, is the call density lower because there's less birds or because it's raining? This is important stuff to figure out, and really have worked into the models. And secondly, I think another thing that we're really curious about is ways to do that covariate analysis in ways that also allow us to reduce the overall validation effort that's needed. Because right now we're very much saying, like, compare broadly comparable things in one way or another, get your call densities for those particular areas, and go forward. But I think as we get some good structure in place for looking at covariates and analyzing covariates and feeding the covariate analysis into, like, where it actually makes the most sense to do the validation effort, then we should be able to get better results with less work.
Nice. I'm also thinking, in terms of some, often or every now and then you see a paper about, ways to standardize our field and have results from one place able to talk to results from another place, and really pool our knowledge. Do you see call density potentially as one of these variables that could be, I guess, shared across studies, or, able to sort of like a standardized metric that maybe comes with some of the metadata from, where you deployed and what equipment you used, and other covariates, but synthesizing this this information to get species populations across much larger spatial scales, like more general population estimates, especially if this becomes an easy to calculate approach similar to like what we've seen with the BirdNet analyzer GUI. Just curious your thoughts on that. Can this be, you know, a relatively standardized metric that could be broadly used within the community?
I certainly think that the call density metric is standardizable. Like we've kind of mentioned, as of right now, there's some considerations about how you subsample and what proportion of the data you're calculating call density for. If you know, one study has 24-hour recordings, whereas another study is only recording snippets throughout the morning, for example, those two call densities are going to be less able to talk to each other, as you said. But I do think that there's an easy way to standardize doing it, and I think it would be really amazing to have a data set across many different ecotypes, many different countries and species, and being able to have this kind of global look at what does call density look like for all of these species. I think that would be really amazing, and I do think it's achievable.
I love that idea. I guess last question on my end. So here you're focused on birds. I would say maybe probably 80% of the BioacousTalks community focuses on species other than birds. And so have you just sort of experimented with, you know, applying this approach to amphibian populations or, you know, insect communities or, you know, in the marine realm, marine mammals? I don't know if Dr. Marquez is on the call, but yeah, I'm just curious what you think the applicability is to non-bird
Yeah,
taxa.
yeah, definitely. I've actually used this protocol for frogs, monitoring frogs on Oahu, an invasive species introduced from Puerto Rico. They're working on control strategies, and we're looking at call density as a measure of how that is going. Not well, by the way. But so it should be applicable for many different vocalizing taxa. Anurans for sure. We've found that embedding space for bird acoustics and Anuran acoustics are actually quite similar. Cetaceans, I have not worked with marine mammals myself. However, our friend at Google, Lauren, she's been working with using this kind of approach with I can't remember what kind of whale it is, but she's been looking at she did some work with whales and found some promising results. So I do think that this is applicable to a large number of taxa. If you wanted to go from call density to animal density, I think it might be a little bit tricky for those species that you can't really sample using more traditional methods. If you can't use, you know, distance sampling on a species, and it might be hard to build these relationships between animal density and call density. But I think as an independent metric, call density offers a lot of utility for those species that can't be monitored using other traditional methods.
Yeah, it could be a great, you know, relative measurement. Well, anybody have any last questions or comments for Amanda or Pat or Tom? If not, mahalo nui for this incredible work that you've been doing. I think, well, certainly I'm going to try this in some of my own work, and I think it hopefully will have ripple effects across Hawaii and then the field more generally. So yeah, really thank you for experimenting and putting in the work to validate this approach and really test it rigorously. And as for us, so this is our last BioacousTalks once again for 2024. We'll be back stronger than ever in 2025, probably starting in March, and then having our spring season beginning. So on the listserv, we'll be sending notifications for upcoming sessions. And if you have any ideas of maybe some research you would like to see featured or ideas for sessions, we're all ears, pun intended. So please let us know. And yeah, just it's been a real pleasure to look forward to these get-togethers every week, seeing people tune in from 30 different countries we've had in some of these sessions, and a lot of you coming back week after week. It's been great to share this space with you and learn about these different bioacoustics works, books, sound installations, whatever, all of it with you. So here's to the last great year we had and another one coming up soon. Take care, everybody. Bye-bye.